Welcome to today's Explainer. We're diving straight into what is, without a doubt, the most high-stakes biological marathon on Earth, human reproduction. We're going to look under the hood at the incredibly complex, completely invisible cellular mechanics that literally build human life. It's an absolute gauntlet of fortified shields and exact chemical locks that had to line up perfectly, just for you to be sitting here watching this right now. Okay, so let's set the stage with a bit of history, because it's actually pretty wild. Back in 1677, when Antoine von Leeuwenhoek first spotted sperm cells, people actually thought they were looking at a tiny, fully preformed human body crammed right into the head of the sperm. No, seriously. It just goes to show how clueless we used to be about this microscopic process. We were basically relying on total guesswork. So, here's our roadmap for today. First, we'll hit that historical mystery. Then, we'll map out the epic microscopic journey. Zoom in on the exact moment of fusion. Look at how we actually track the pregnancy timeline. And finally, wrap up with the barriers that can stop this whole process in its tracks. Section 1. The historical mystery. From ancient guesses to microscopic truths. For the longest time, going all the way back to antiquity with guys like Hippocrates and Aristotle, it was all just philosophical debates about where babies came from. And consider the timeline gap here. Even after discovering sperm in 1677, it took until 1827 for Carl Ernst von Baer to actually observe a human egg. That is a solid 2 centuries of squinting through early microscopes until Oscar Hertwig finally proved in 1876 that fertilization is the literal fusion of an egg and a sperm cell. Centuries of mystery finally solved. Section 2. The epic microscopic journey. A high-stakes marathon. People often talk about this like it's a simple swimming race, but it's really not. Think about the massive geographic scale here. Fertilization doesn't just happen anywhere. It primarily goes down way up in the ampulla of the fallopian tube, right where it curves around the ovary. The sperm has to trek all the way from the uterus up into these incredibly narrow, specific tubes. And the attrition rate? It's absolutely staggering. Out of a starting lineup of about 200 million spermatozoa, guess how many make it to the ampulla? Just 200. 200 elite survivors out of millions. They're swimming at maybe 1 to 3 millimeters a minute, entirely against the current. It is a brutally hard trek. But here's the thing. The egg isn't just sitting there passively waiting. The cells around the oocytes secrete progesterone, which acts like a massive chemical beacon. This progesterone binds to receptors on the sperm, spikes their internal calcium, and triggers this hyperactive swimming mode. Basically, the sperm just follows the chemical trail, which guides it straight to the target. Section 3. The moment of fusion, breaching the fortress. Just getting to the egg is only half the battle. Now, the winning sperm has to break through some seriously heavy defenses. It goes through intense physiological changes to pull this off. First is capacitation, which hyperpolarizes the membrane. Then it pushes through the corona radiata, that outer layer of cells. Finally, it hits the zona pellucida, this tough glycoprotein shell. And right there, the sperm's cap, the acrosome, literally bursts, letting out an enzymatic explosion to melt right through the armor. As the sequential phases of fertilization play out, the sperm touches that gelatinous envelope, releases its enzymes to dissolve a path, and finally buries its head deep enough to fuse its membrane directly with the egg cell. It's basically a highly coordinated cellular break-in. Okay, wait, here's a truly mind-bending fact. Some sperm cells will actually burst their enzymes way too early on the egg's surface, effectively destroying their own chances of fertilizing it. Why would they do that? Well, sperm from the same cohort share about 50% of their genes. By blowing up early, they weaken the egg's defenses so a closely related brother sperm can get through. It's literally a microscopic version of kin selection, taking one for the team. But the second one sperm successfully fuses, the game is over. The egg instantly triggers what's called the cortical reaction. It is an absolute immediate lockdown. Why? Because if multiple sperm got in, a condition known as polyspermy, it would cause a lethal offset of chromosomes. Right inside the egg's membrane, there are these little cortical granules just waiting. The moment fusion happens, they expel their enzymes outward. This causes the surrounding matrix to cross-link, instantly turning into a rock-hard, impermeable chemical shield. Nobody else is getting in. Inside, we get this incredible genetic handshake.
A sperm protein, literally called izumo, binds perfectly with a Juno receptor on the egg. It's a lock-and-key fit. After that, the sperm's tail degenerates, their membranes dissolve, and they go through their first mitosis. This is the exact moment those 23 chromosomes from the sperm and 23 from the egg truly fuse into one unique entity. The blueprint is officially complete. Section 4. The pregnancy timeline. 2 ways to keep time. So, once you have this zygote, how do doctors actually track its age? Well, there are 2 distinct ways, and it causes a lot of confusion. Gestational age measures from the start of the mother's last menstrual period, but fertilization age is measured from the actual moment of conception we just talked about. Medically, gestational age adds about 14 days to the fertilization age. So yeah, expecting parents are often pretty confused when the doctor's math doesn't match the exact date they know they conceived. When you stack these medical milestones up side by side, it's pretty wild to see the difference. When the embryo stage kicks off, the fertilization age is only day 15. But medically, the gestational age is logged at 4 weeks. By the time the fetal stage begins, you're at 8 weeks fertilization age, but over 10 weeks gestational. Now, during those critical first few days of the fertilization age, things move fast. The fertilized oocyte starts splitting, 2 cells, then 4, then an 8-cell morula, all while continuing to roll down the fallopian tube. Eventually, it becomes a blastocyte of about 70 to 100 cells, drops into the uterus, and finally implants. Section 5. Barriers to fertility when the marathon is blocked. Of course, biology is messy, and this highly intricate marathon doesn't always go according to plan. There are plenty of roadblocks. Like we mentioned, polyspermy is lethal, but you also have conditions like PCOS, which can delay or completely stop ovulation. Autoimmune disorders might cause the body to reject an embryo. Endocrine issues like diabetes or thyroid problems can mess with those vital chemical beacons we talked about. And things like endometriosis can create severe physical barricades that just stop the journey outright. So, taking a step back, when you consider the sheer scale of all this, 200 million competitors, a treacherous microscopic trek, heavily fortified biological shields, sacrificial cellular brothers, and exact protein locks that must perfectly align, it really makes you wonder, doesn't it? The start of human life is just so astonishingly precarious and mathematically improbable. Frankly, it's an absolute miracle any of us are here at all. Thanks for hanging out with me on this explainer, and keep exploring.